We'll get this show on the road. Well, thank you everybody for coming this morning to Stephen Gurney's Master's Thesis Defense. I'm his graduate supervisor, Sonia Christensen. Stephen is my first graduate student, so it's very exciting. I want to say thank you to him right off the bat for taking a chance on me, and actually didn't take that much of a chance on him because I knew him since he was an undergrad when he was doing some bear research for his undergraduate research project. And he was a FW graduate here at MSU, then went off to do a variety of tech jobs, came back, started working on the project that he's going to be talking about today and the first field season in 2019. And then he stuck around another field season and then stayed on as a grad student. He's also the only grad student on this project for a 4-year field project. We had a lot of other good teammates, Melissa, helpers from the DNR, lots of great collaborators from the DNR and others who have really made this project happen. But again, I just really want to highlight Stephen here. He also took a little bit of a circuitous route before he got to FW. He was roofing beforehand and decided that, you know, as good of a life as roofing was, he wanted to pursue his dream and wildlife management and conservation. So when he came this way eventually, and so happy that he did. So with that said, I'll let Stephen take it away here. He's going to be recording this, and we've got some folks online. I'll be managing this, and yeah, we'll let him know.
Thanks for that, Sonia. Hello, everybody. Thanks for coming out and hanging out with me this morning. The title of my thesis is Navigating Uncertainty in Wildlife Management, an empirical evaluation of a regulation change and subsequent impacts on a deer population. The focal species was white-tailed deer, and the harvest regulation change evaluated was the implementation of antler point restrictions. And this whole study was in the context of chronic wasting disease. So the central question of this research was, how does a harvest regulation change affect a deer population? And the central theme of this work was navigating uncertainty. So first, observational uncertainty, which arises because the state of the system must be inferred from imperfect monitoring systems. And second, structural uncertainty, which represents our imperfect knowledge about how a managed system behaves. So let me give you a quick broad overview of what I'll be talking about today. In chapter 1, I addressed observational uncertainty by evaluating an abundance estimator. And as a result, I identified camera trap sampling strategies for precise and reliable abundance estimation. So the work from chapter 1 informed modeling efforts in chapter 2, where I addressed the structural uncertainty by measuring a population change following a harvest regulation change. And what I found was weak evidence for the regulation change increasing adult male abundance, and I found no evidence for the regulation change decreasing overall deer abundance. So why do we care about deer and deer harvest? Well, from a management and conservation perspective, deer are a very important species. As herbivores, they play a critical role in the ecosystem. They are appreciated by wildlife viewers, and they have intrinsic value as a game species. And that's really important because deer hunting related expenditures and license sales generate a substantial portion of conservation funds, not only for the benefit of game species, but for non-game too. So for these reasons, wildlife managers must assure the health of free-ranging deer populations. But we got a big problem, chronic wasting disease, also known as CWD. CWD affects cervids or species in the deer family, and once a deer is infected, it is always fatal. The disease agent is a misfolded protein called a prion, depicted here in the top right. CWD is not only transmissible through animal-to-animal contact, but it also acts as an environmental contaminant, which can further contribute to the maintenance of the disease. In areas where CWD is established, it has emerged as a major threat, reducing the health of wild cervid populations and causing long-term population declines. It can also discourage hunters from harvesting deer in and around affected areas. So consequently, CWD jeopardizes wildlife conservation and management more broadly. It's not just a deer issue. And it's been described as one of the most challenging wildlife diseases to manage. One unique challenge is that CWD affects sex and age classes disproportionately. Here's a hypothetical example, just for visual purposes. Depicted left to right, we have a fawn yearling female. In the middle with the small antlers, we have a yearling male, then an adult female, and on the far right, with the larger antlers, we have an adult male. So prevalence generally increases with age, which is a likely consequence of an increase in human infection risk. And prevalence is estimated to be about twice as high in adult males than females, which is likely due to differences in behavior. For example, yearling males disperse at the greatest frequency within white-tailed deer populations, and mature males increase their movements and expand their home ranges seasonally. And both of these behaviors can contribute to the spread of disease. Now, zooming out to talk about disease on a larger scale, this map depicts the distribution of CWD in North America about 10 years ago. Gray counties representing CWD and free-ranging populations. So these data are from 2013, and fast forward to today, and you can see that in just 10 short years, CWD has been detected in many new areas. So needless to say, CWD is becoming more relevant every day. And so how do we fight this disease? Well, one of the most powerful tools wildlife managers have in their toolbox is control over harvest regulations. Both population management and disease management are highly dependent on harvest. And state agencies often employ sex and age-based harvest criteria. And when that criteria is changed, it's expected to exert a desired population effect. This matters because host population size and structure can have implications for disease transmission. And following the detection of CWD, agencies generally alter deer harvest regulations or initiate management practices in an attempt to reduce disease transmission. Some studies have shown that reducing deer densities has been successful at maintaining low prevalence of CWD, and most of those density reduction programs focus on female harvests to reduce a population's reproductive potential. Other studies have considered harvest strategies that target specific high-risk sex and age classes as a useful approach for CWD management. So effective harvest programs also require consideration of hunter preferences, and many hunters prefer to harvest older male deer with larger antlers. And mandatory antler point restrictions, or APRs, are an example of a deer harvest regulation expected to change the male age structure of deer populations. Specifically, APRs are designed to protect younger antlered males from harvest, improve their survival, and thus increase recruitment into the adult age class. There are many types of APRs, but a common one is the 4-point restriction. So what that means is that hunters may only harvest antlered deer with 4 or more points on a single antler. And since younger males tend to have smaller antlers, a 4-point restriction protects most yearling males from harvest. So in recent years, there's been a growing interest in antler point restrictions. So we got chronic wasting disease becoming more relevant, and we got antler point restrictions becoming more popular. But natural resource agencies have limited information on how APRs impact deer populations. So this study was designed to evaluate the impact of an APR regulation change on a deer population in Michigan's CWD core area. It included a statistically robust before-after controlled impact study design with an APR treatment in the eastern 3 counties here in green and a non-APR control area in the western 2 counties here in brown. Sampling occurred at the township level across 8 townships total, here in light gray. And these townships, they were systematically selected and paired between APR and non-APR zones. Pairing criteria included similar geographic features like rivers, similar land cover composition like percent forest, and similar amounts of public hunting land. So for an example, Township Pair A was characterized as heavily forested with large portions of public land, and Township Pair C was agriculturally dominated. This study included a camera trap sampling design, and sites were selected randomly at the township section scale using a generalized random tessellation stratified approach. Camera sites were non-baited, and to minimize bias, we deployed 144 motion-activated infrared camera traps with 18 cameras per township, and those cameras were set to take bursts of 3 photos with 5-second delay between bursts. Photo data was collected during what was considered a 9-week closure period during summer months in which deer movement was limited. So it was assumed that no individuals were leaving or entering the population. And surveys were conducted from July 15th to September 15th for 4 consecutive years. All photos of deer were characterized based on the number of individuals in the frame, sex and age class of individuals, and the number of antler points. We had dual observers and an expert judge to resolve any discrepancies. And in the later years of the study, an artificial intelligence software megadetector was used to accelerate photo processing. In the end, we collected far more images than we had anticipated, over 2.6 million photos total. So this high volume of images presented a great opportunity to perform a methods assessment, and chapter 1 is a methods assessment. So here in chapter 1, I address observational uncertainty by examining how camera trapping strategies affect deer abundance estimates. And this included empirical data and simulation-based approaches. So population abundance measures are important to ecological studies, and they form the foundation of effective wildlife monitoring programs. Whether studying threatened species, invasive species, or game species, an understanding of abundance is important for evaluating the impact of conservation and management actions. However, obtaining reliable abundance estimates can be logistically difficult, time-consuming, and costly. And given the limited resources available for monitoring wildlife populations, there's a critical need for reliable and cost-effective methods sensitive enough to detect population trends. Today, camera trapping is a standard method for monitoring wildlife populations, and where camera traps really excel is collecting count data for unmarked animals across large spatial scales. When I say unmarked, I mean individuals are not distinguishable. There now exists a suite of models that leverage such data for estimating abundance. So the surge in popularity with camera trapping, combined with improvements in technology and data management, often results in the accumulation of large data sets. And this really exposes the Achilles heel of camera traps. So it's an issue that's becoming more relevant, which is the labor required to review high numbers of photographs. So what this does is it forces wildlife managers to compromise between the camera trap sampling strategy and data quality or reliability of the abundance estimates. So this is quite the balancing act, and it raises the big question, how much sampling is enough? So to answer this question, I took advantage of a camera trapping data set from the larger APR study and used these data to address the missing piece, which was, we don't know how sensitive abundance estimates are to alternative sampling strategies. So here with chapter 1, I helped fill this knowledge gap, and I did so by addressing these 3 objectives. I evaluated the sensitivity of abundance estimation to sampling frequency, to sampling duration, and to camera distribution. It's important to note that these 3 sampling strategies are vital aspects of camera trap study design. And here's how I did it. To model abundance and account for imperfect detection, I used a closed in-mixture model. My modeling decisions were informed by another effort, an exploratory data analysis, where I evaluated in-mixture models with alternative mixing distributions, including Poisson, zero inflated Poisson, negative binomial, and Poisson log normal. I also evaluated potential covariates for detection probability, including near and far cover, canopy cover, cover type, proximity to open water and wildlife trails, presence of deer sign, field of view size, and multiple variants of time. Abundance covariates explored included dominant land cover types as percent forest and percent agriculture. I modeled abundance using a Bayesian implementation using JAGS and Program R. I used vague priors, and all sex and age classes were modeled together to produce a single study area estimate. So essentially, the in-mixture model consisted of 2 linked generalized linear models. The ecological process model yields the latent state and the spatial variation of abundance at site I for a group of sites distributed by a Poisson distribution with a mean lambda. I use a log-link function to incorporate a covariate described by a nonlinear relationship with percent agriculture cover as a site-specific covariate, and a random intercept kappa, which allowed township to act as a random effect on abundance. Next, the observational process model yields the observed counts Y at site I during replicate survey J, distributed by a binomial distribution with sample size N and detection probability P. I use a logit link function to incorporate a covariate distributed by a linear relationship, described by a linear relationship with site-specific cover, open or closed. And it's important to note that my abundance estimates reflect relative abundance, not absolute. So I'm estimating abundance for a population associated with a smaller geographic area. And since truth was unknown in my study, I used a modeling approach to explicitly simulate true scenarios of deer abundance. And these simulations allowed me to investigate the performance and accuracy of my in-mixture models. I incorporated parameters estimated from the empirical data, including coefficient values and mean site abundance by township. And for each set of simulations, I defined the scenario-specific daily sampling frequency, sampling duration, and camera distribution. And for each scenario, I generated 50 unique data sets to capture variability and random draws. I used root mean square error as a measure of overall accuracy, complemented by relative bias analysis to determine directionality of any bias. I aggregated posterior distributions from the 50 unique data sets generated and summarized mean and 95% quantiles. And to reduce computation time, I used supercomputing resources at Michigan State University's High Performance Computing Center. So now talking about how I modified sampling strategies, starting with daily sampling frequency, or how often we count deer. I restricted counts to the maximum number of individuals in a single image per unit of time. And to better align with deer activity, I rescaled standard time to local solar time and counted deer once per day, counted deer twice per day with midday at solar noon, I counted deer 3 times per day, 4 times per day, and 2 crepuscular counts per day with 2-hour windows around sunrise and sunset. I also simulated reductions in sampling duration. I did this by reducing the number of days in the sampling period by 7-day steps from the full 63-day sampling period. Those days were selected at random. So I used a 56-day sampling duration. I used 49 days, 42 days, and 35 days. I produced 10 unique data sets per scenario, aggregated them, and summarized mean and 95% quantiles. I also modified camera distribution, or the number of camera sites per township. And I simulated reductions by steps of 3 from the full 18-site distribution. Those sites were selected at random. So I simulated 15 camera sites per township, 12 sites per township, and 9 sites per township. Again, 10 unique data sets were generated and aggregated for each scenario, and I summarized mean and 95% quantiles. Moving on to results now, starting with the photo data that I used. I used a subsample of data from 2019, which was about 800,000 images, including 276,000 images of deer. Does or female deer were photographed most, and legal males or antlered deer that met the APR criteria were photographed the least. Here's when photos were taken. Got time of day in the x-axis, deer detections on the y, and sex and age class by color. I found that all sex and age classes followed a similar activity pattern, with most detections around sunrise and sunset. So this is important to consider when thinking about sampling frequency. Moving on to the sensitivity analyses, starting with relative abundance here on the y-axis, sampling strategies on the x. Now the data. Looking at daily sampling frequency, I found that mean relative abundance estimates generally increased as sampling frequency increased. But I did have one exception with crepuscular sampling, producing abundance estimates significantly lower than all other scenarios. Sampling duration also had a significant effect. There's a positive relationship between the number of days in the sampling period and relative abundance. With 56-day and 63-day scenarios producing estimates significantly higher than the 35-day scenario. But overall, relative abundance was most sensitive to modifications in camera distribution. And as expected, I found that relative abundance estimates decreased as the number of camera sites decreased, with most significantly different from one another. And this relationship can be explained by abundance being associated with a smaller geographic area. Now for the precision of those abundance estimates, I use the coefficient of variation to facilitate comparisons. Here on the y-axis expressed as a percent, with lower CV values indicating higher precision. I found that precision was relatively insensitive to changes in sampling frequency, with CVs approximating 3% for all scenarios. In comparison, precision was more sensitive to sampling duration and slightly improved as the length of sampling period increased. And precision was most sensitive to camera distribution and improved as the number of camera sites increased. Notably, the 9-site scenario produced the least precise estimates, with a CV 2.6 times greater than the 18-site scenario. So these results highlight the in-mixture model's ability to produce relative abundance estimates with high precision. CV less than 25% is often considered sufficient for rough wildlife management decisions, and less than 12.8% for precise management decisions. And here my results range from about 3 to 9%, and compared with other similar studies, this level of precision was higher than previously recorded in the literature. Moving on to detection probability. I found that detection probability was most sensitive to daily sampling frequency. Detection probability decreased as sampling frequency increased, with the 1 per day scenario producing significantly higher detection probabilities than all other scenarios. Interestingly, crepuscular sampling produced detection probabilities comparable to the other scenarios. And given these results, it's likely that the lower abundance estimates observed with the crepuscular sampling were driven by missed counts. I found an inverse relationship between the number of days in the sampling period and mean detection probability, but estimates were comparable. Overall, detection probability was relatively insensitive to changes in camera distribution, but uncertainty grew as sites decreased. So this study aimed to disentangle strengths and weaknesses of alternative camera trap sampling strategies, and low detectability was recognized as a common limitation. However, it's not uncommon to have detection probabilities less than 0.5, and given that detection probability increased when I decreased daily sampling frequency, my results support a coarser sampling frequency as a viable option for increasing detection probability. Using a simulation-based approach, I also evaluated the sensitivity of model performance, root mean square error here on the y-axis, which relates to the number of deer at a camera site. The big takeaway across these three is that the model was able to predict site abundance within about 3 deer per site. I also evaluated the relative bias here on the y-axis to determine any direction of bias. Here indicated by the red dashed line, mean estimates approximated 0 and error was centered around 0, which indicated no bias between simulated truth and predicted abundance. So to reiterate, the model was accurate. It was able to predict abundance within about 3 deer per site, and estimates were not biased high or low. So what does this all mean for resource savings? Well, altogether, the collection, management, and processing of image data required an estimated 4,700 person hours of work in a single season, so just one season, and a large portion of photo processing time was spent reviewing irrelevant images or images without animals. Taking a look at this histogram here, time of day on the X, number of photos on the Y, brown corresponds to photos with deer, tan without deer, and those distributions are overlaid with transparency. What should really jump out here is the mountain of photos without deer. Excluding this midday period would translate to a whopping 1,500-hour reduction in photo processing labor. And although these results here focus on white-tailed deer, they really highlight the importance of sampling relevant to the activity of any study species. Alternatively, a machine learning application could be used to accelerate photo processing by filtering out images that do not contain animals. So 4 big take-home messages from chapter 1. Number 1, our results demonstrate that irrespective of data modifications, in-mixture models can perform well for the relative abundance estimation of white-tailed deer in the south central lower peninsula of Michigan based on the modeling of camera trap count data. 2, in-mixture models should be paired with careful consideration of assumptions and sampling strategies, as large uncertainty and parameter estimates can obscure signals of change in abundance over time and space. 3, shorter sampling durations and lower camera distributions can facilitate resource savings, but my results showed reductions in sampling intensity compromised the precision of abundance estimates. And lastly, my results highlighted the importance of designing sampling efforts relevant to animal activity and its potential for application in improving sampling efficiency. So working towards answering that bigger question, Chapter 1 helped inform modeling efforts in Chapter 2. Recommendations included the use of a weekly count for sampling frequency to improve detection probability, to use 100% of the data to maximize precision of abundance estimates, and to use artificial intelligence software to help accelerate photo processing. So in Chapter 2, I addressed structural uncertainty by assessing the effect of a deer harvest regulation change on relative abundance using a before-after controlled impact study design. So assessing changes in population abundance is important when evaluating the effectiveness of conservation and management of wildlife. However, evaluating a single regulation change on a free-ranging population is considerably challenging, and robust evaluations generally require coupling the regulation change with before and after population monitoring within treatment and control areas. For game species, many agencies instead rely on estimated or enumerated harvest for population monitoring. Although useful, the sex and age data of the harvested population may be non-representative of the true state of the population. For example, changes in harvest trends may reflect changes in hunter effort or changes in harvest regulations rather than true population level changes. Ultimately, wildlife management actions cannot be thoroughly assessed without reliable data on population parameters. So mandatory antler farm restrictions are an example of a harvest regulation frequently applied to white-tailed deer management programs. In theory, implementing an APR that protects most yearling males from harvest will result in increased yearling male survival, and thus increased recruitment of yearling males into the adult age class. APRs have been implemented and evaluated under a diversity of conditions, but have been found to successfully reduce the proportion of yearling males in the antlered harvest. Numerous studies have also observed increased proportions of older antlered deer harvested following APR implementation, but these increased proportions may be explained in part by lower proportions of yearling male harvest. So a prevalent hypothesis is that APRs increase the abundance of adult males in a population. And given the direct relationship between antler growth, age-specific male survival, and male age structure, this hypothesis has foundations in logic. However, past studies have lacked estimates of male abundance by age, thus it was not possible to evaluate the magnitude of effect APRs had on adult male abundance. So APRs reduce the availability of harvestable yearling antlered deer, which may lead hunters to shift harvest pressure to antlerless deer. And the additional compensatory harvest pressure on antlerless deer can translate to an increased female mortality and a subsequent reduction in a population's reproductive potential. Collectively, these ideas have led to another prevalent APR hypothesis that APRs decrease overall deer abundance. To my knowledge, only one study supports this hypothesis, but it was the only study in the literature to explicitly evaluate it. Overall, there's a lack of information on the effects of APRs on overall deer abundance. So despite the widespread application and increasing interest of APRs, a critical knowledge gap remains, which is we don't know how APRs affect deer populations out there on the landscape. Do APRs increase the abundance of adult males? Do they reduce overall deer abundance on the landscape? Most APR studies to date have focused on evaluating harvest in response to APR regulation changes. So consequently, population level effects of APR remain unclear, particularly on sex and age distribution, warranting empirical investigations. Which leads me to our case study, which I mentioned earlier. Back in 2019, the Natural Resources Commission implemented APRs in the eastern half of the CWD core area. And our research was coupled with this harvest regulation change. And I want to emphasize that opportunities to pair research with a regulation change are few and far between. And this makes our study unique to most APR studies to date. And that was all made possible by a close collaboration between MSU, the Michigan Department of Natural Resources, and the Natural Resources Commission. It's important to note that camera trap surveys were conducted each year before harvest occurred, And to reiterate, this is a pre-post APR study. It includes 1 year of data before the regulation change and 3 years of data after the regulation change. So pairing the regulation change with research provided me an opportunity to empirically evaluate these two prevalent independent APR hypotheses. And if the hypothesis about APR's increasing adult male abundance is true, you would expect to see abundance of adult males increasing over time in the APR zone with no changes in the non-APR zone. And if the hypothesis about APR's decreasing overall deer abundance is true, we'd expect to see the abundance of other sex and age classes decrease in the APR zone with no changes in the non-APR zone. So here in chapter 2, I evaluated these two hypotheses, and I did so by estimating annual relative abundance using year stratified admixture models. Different from chapter 1, these are open population hierarchical models, and they are sex and age specific. And to fit these models, I converted from JAGs to Nimble for increased flexibility and efficiency. I used several approaches to interpret the effects of harvest treatment on deer abundance. I compared relative abundance estimates over time, I visualized marginal effects, and I quantified changes in abundance. Models here extended upon the static binomial admixture model used in chapter 1, and some of those improvements included addressing spatial temporal differences in the data, which reduced noise around the estimates. There was also information sharing across years, which really unlocked the full potential of the data. And where it really shines is that the year stratified model is specifically designed to evaluate trends or covariate effects. So it explicitly accounts for the effects of harvest treatment year and the interaction of the two. And here's what that looks like. Compared to the model described in chapter 1, the year stratified model had an additional level of hierarchy, T for year, highlighted in yellow if you can see it. Also new and important to the hypothesis testing, highlighted in yellow here, are the beta 1 year specific intercepts by harvest treatment and beta 2 year specific interactions between year and harvest treatment. I also included a township random effect modeled as a residual term epsilon. Essentially, the random effect gave the model latitude to account for variation in mean abundance among township and year combinations. Note that harvest treatment was not modeled as a binary covariate via categorical indicator variables, but instead as categorical index variables. So simply put, I had 2 values for the intercept and 2 values for the interaction term, which the model assessed by referencing the specified index variable, APR or non-APR. And this mechanism was advantageous over common indicator variables, like using 1s and 0s as on-off switches, which can introduce unwanted asymmetric uncertainty between treatment groups. Used a similar indexing structure to introduce the township random effect. The observational process model also included the additional level of hierarchy for year, and a new random effect modeled as a residual error term epsilon, which gave the model latitude to account for heterogeneity and detection probability among sites and years, which, if not addressed, would confound trend estimates. So as a reminder, I'm estimating relative abundance, and building off this framework nicely described by Gilbert et al., each camera viewshed here in blue is associated with a green effective sampling area around it. And note that the effective sampling areas are functions of animal movement and survey length. So I produce relative abundance estimates for each green effective sampling area or site. And I also aggregate those site estimates within the orange sampling frame to produce relative abundance estimates by township. And so when I say relative, I mean relative to the before-after control impact study design. So for an example, township abundance in the treatment area relative to township abundance in the control area, or township abundance in 1 year relative to township abundance in another year. So I considered a minimum of 3 years post-harvest regulation change was needed to observe and evaluate potential population level changes. The males, including fawns, that remain after harvest need time to mature and advance to older age classes, and any change in antlerless harvest pressure needs time to affect the female population, fawn production, and maturing of individuals. Simply put, comparisons between 2019 and 2022 are really important. Now moving on to the results here, starting with township level relative abundance. Here, abundance of legal antlered deer on the y-axis, year on the x. In the left column, we have the APR zone, non-APR zone in the right. And in the rows, we have township pairs side by side, township pairs A through D. And as a reminder, 2019 estimates represent baseline estimates for the system before the APR regulation change. And note that due to COVID-related constraints, only half of the paired townships were surveyed in 2020. Adding the data, relative to 2019 baseline estimates, I failed to detect any significant changes in APR or non-APR townships. But in general, mean values increased over time in both zones. So these similar increasing trends, they do not strongly reflect what was hypothesized. Now looking at relative abundance of female deer, relative to 2019 baseline estimates, I detected a single significant increase in the non-APR zone in 2021, but it did stabilize the following year. As it relates to the hypothesis being tested, it is not reflected here with these results. Although there is some noise around the data, but big picture, female abundance is not decreasing significantly anywhere. Moving on to sublegal antler deer. Relative to 2019 baseline estimates, I detected increases in the non-APR zone in 2022 in Township A and Township D. And although not significant here, mean values generally increased over time in both zones. Again, these results do not reflect what was hypothesized. Lastly, fawn relative abundance. Not much happening here, things relatively flat and not reflecting the hypothesis. No significant declines in fawn abundance anywhere, which is not surprising because female abundance was not declining either. So it can be difficult to interpret results when there's noise around the data, like there was with some of my township level abundance estimates. So I use model predictions to help with interpretation here. I visualize marginal effects to strip away some of that noise and to help interpret the abundance change over time. On the y-axis here, we have the effect expressed in terms of mean site abundance, so at the site level. Year on the x-axis, township pairs in separate panels, and harvest treatment by color. First up, we have legal antler deer. So here we're just visually inspecting the plot and interpreting slope trends to help determine whether the implementation of APRs produce a negative or positive impact on deer abundance. So error bars are less important here. But what should really jump out is positive trends and marginal effects across all townships. So the model predicted that abundance was increasing over time regardless of harvest treatment. And those slopes look pretty similar to one another. We saw a similar pattern with female deer, slightly smaller scale here, but slopes almost perpendicular to one another. We saw similar increasing patterns with sublegal antler deer. And although very small, the model predicted increases in fawn abundance regardless of harvest treatment. So we already looked at relative abundance at the township scale, and we used marginal effects to help interpret those results. Now, another way to interpret abundance change over time is to compute the difference between difference in abundance from the first year to the last year of the study. So that's what we're looking at here. It's further simplified and a finer resolution. So relative abundance is now on the site scale. Here on the y-axis, we have the mean change in site abundance township pairs on the X, harvest treatment differentiated by color, and the horizontal dash line at 0 indicates 0 change. Adding the data, township pair credible intervals overlapped one another, and no credible intervals overlap 0. Simply put, from the start to the end of the study, abundance increased similarly among APR and non-APR townships. Note that mean values approximated an increase of 1 deer per site, and mean values were slightly higher in the APR zone. Now looking at female deer, notice a smaller scale here. Magnitudes of change were similar between paired townships, and all credible intervals overlapped 0, which indicated there were no significant changes in site abundance. However, posterior distributions included mostly positive values, suggesting a general increasing trend. Moving on to sublegal antlered deer. Township pair credible intervals overlapped, and it was close, but no credible intervals overlapped 0. So put another way, abundance increased similarly among APR and non-APR townships, with average increases less than 1 deer per site. And lastly, we have fawns. Similar results among paired townships, and I failed to detect any significant changes. Note that mean values here were all less than 0.5, but posterior distributions included mostly positive values, suggesting a general increasing trend. So, some key discussion points. I found weak support for APR's increasing abundance of legal antlered deer. Surprisingly, I found similar increases between APR and non-APR harvest treatments. So why is this? Well, it's likely that voluntary APRs played a role in results, and partial controllability of hunter harvest can limit the influence of management decisions on realized harvest. And although evaluating harvest data was not an objective of this study, it's important to consider here. In the decade leading up to this study, yearling male harvest declined by about 10% in the study area, decline depicted here, year on the x-axis, percent of antlered deer on the y. Further, prior to this study, surveys indicated that 63% of hunters in the study area supported the implementation of these APRs. And in 2022, the state started a mandatory harvest reporting program. And looking at those data, most antlered deer harvested today meet APR criteria, which suggests a statewide trend for hunter selection for larger antlered deer and self-imposed restrictions on yearling male harvest. On this heat map here, that black county on the bottom indicated 80 to 90% of antlered deer harvested met the 4-point APR criteria. Dark gray was 70 to 80%, and so on. So looping way back to chronic wasting disease, hunter selectivity for older deer with larger antler growth has implications for CWD spread and growth. This type of selective behavior may influence the size of the adult male population on the landscape, as suggested by this study. And from a CWD management perspective, this is concerning, given that CWD prevalence generally increases with age and is highest in adult males. In another effort, I used empirical data from this study and informed and adapted a published agent-based model. And that published model I used was Michigan-specific and designed to simulate CWD spread and growth. So using data from the study, what I found was that when there were higher proportions of adult males in the antlered population, there were more severe CWD outbreaks. Looking at plots here, working left to right, when the antlered population had higher proportions of antlered males, there was a significantly higher number of CWD cases, more CWD area, and higher CWD prevalence. As it relates to the other hypothesis tested, I found no support for APRs decreasing overall deer abundance. So why was that? Well, the hypothesis is dependent on augmented antlerless harvest, which did not materialize. Looking at harvest data here from the study area, antlerless harvest on the Y, year on the X, we see that antlerless harvest was similar between APR and non-APR counties during the study period. 2020 was a unique year with statewide increases in hunter participation in antlerless harvest, which likely reflected the effect of COVID-19 restrictions, allowing additional people to hunt and additional antlerless harvest opportunities that were available in the lower peninsula. And in 2020, counties in the study area accounted for some of the highest antlerless harvest densities in the state of Michigan. However, despite the increased mortality of antlerless deer, I failed to detect any significant declines in female relative abundance in 2021, boxed out, shaded in gray here. Actually, most mean values were higher in 2021 than 2020, suggesting that the population continued to grow despite higher hunter participation and higher antlerless harvest. So some big takeaways from chapter 2. Number 1, I found weak support for the hypothesis that APRs increase adult male abundance. Both APR and non-APR treatment areas, I consistently observed increasing trends in relative abundance of legal antler deer, and the magnitudes of change were unimportant at the deer management scale. Number 2, I found no support for APRs decreasing overall deer abundance. Actually, I found that deer populations were increasing within the study system, regardless of harvest treatment, and this was further supported by model predictions. Specifically, these results highlight potential limitations of recreational harvest achieving desired effects for population reduction goals, at least under current regulatory systems. My results also highlighted the importance of developing harvest regulations specific to the targeted deer population, area, and management goals, and I recommend careful consideration of the partial controllability of harvest regulations. So chapter 2 helped address the larger management question, and the findings from this chapter were very well supported and can be used to help managers and hunters set reasonable expectations for changes in deer abundance by sex and age class under antler point restrictions. And lastly, I applaud the Michigan Department of Natural Resources and the Natural Resources Commission for making this study a reality. I want to thank my advisor, Dr. Sonia Christensen, my committee members, Rick Campa, Scott Winterstein, Sarah Mayhew. Thanks all my collaborators at MDNR, including Dwayne Etter, Melissa Nichols, Chad Stewart, Mario Liberati. Big thanks to Neil Gilbert with the Zipkin Lab, and David Williams and Rose Stewart from the former QWC. Many thanks to the many partners that made the study possible, including 160 private landowners. Big thanks to the army of people that helped collect and process data. And lastly, I'd like to acknowledge sponsors of this work. And if I got time, I'll take some questions.
Hey, any questions for Stephen? We've got plenty of time.
Yeah.
So, Steve, you did a really nice job laying out the hypotheses and what you thought you'd see based on previous studies. And I just wonder if those other studies had the numbers of deer that we do in southern Michigan. So I kind of wonder if the overabundant deer in southern Michigan kind of overwhelm any type of regulatory change, that the effect of a regulatory change that was maybe observed in other studies. So you got any thoughts on that?
So the question was, is there any numbers out there that looking at deer numbers and harvest and how it could affect the population? So I guess the only one other study that's looking at that explicitly looked at how APRs are influencing population size was the one in Pennsylvania. Yeah, I think southern Michigan's, that agro-forested dominates, it's a deer mecca over here. Yeah, it probably does have something to do with it, definitely.
Yeah. I just, you wonder about, there's just so few deer, relatively speaking, there's so few deer harvested to what's available on the landscape. It probably plays a role in your lack of detecting some of the patterns you expected to see, right, in the hypothesis form. That's something I was thinking of as you were presenting.
Yeah, good point. Yeah. Thanks. Do you have a question?
Yeah, got it. So, question. I kind of raising it up a bit for something like that. The effect of COVID, I think, is fascinating. This is sort of an actual experiment for one of our assessment question. So I was wondering, based on what Gary was saying, do you think that you just couldn't see the effect that you were expecting because COVID is having all these multitudinous effects on the population of deer at large, so yeah, you get some increased hunter participation, but maybe all they were doing was harvesting those deer that were most likely to be harvested during that particular year anyway. And you were bumping deer, all the way across the landscape. You have basically an open experiment, there you have a lot of immigration and immigration, and deer that are all over the place. I'm just wondering if you think that the study would benefit long term from maybe a comparison during a period of time when you don't have as large an effect as COVID, or are you able to control for that in some way?
Yeah, so the question was, would it be good to have a study to compare to that, like where COVID doesn't happen? Yeah, so the before-after control impacts study design. We try to control everything that we possibly can within the framework, but a lot of things are going to be out of our control. And yeah, I think maybe the COVID, it did play a role in the results, maybe. So hard to tell, but it would definitely be worth comparing. Sure, let's do it again. Yeah.
All right, I have two questions. I'm going to try and tie up some very good comments for the second chapter, and I'm going to ask the question on the first chapter. But your model structure in the second chapter sort of implicitly assumes that there's an instantaneous response to the density, right? There's no population lag that you would have in an integrated population model. So my question is, how variable do you expect your populations to be in general through time? Because that would imply limited power if they would detect a change.
Yeah, there's going to be natural variation there, year to year. I do think that it's super difficult, and is it even possible to detect change at that small of a scale?
Well, especially when you think about the fact that you have 2019 measurement, you said it's compared to 2022 measurement. You got a lot of fancy math, and I like Bayesian models, but you have an N of 1 for that particular comparison, right? So that, I guess that was the point. But I think it's related to Gary's point earlier. And then the second question was for chapter 1. You showed that the simulation return, the model is estimated really nicely. Were you using the exact same simulation model as the estimation model? Assuming you were,
yes.
Vague priors.
Yes. Okay,
so what you were showing there was, I know it's Bayesian, but you're showing consistency of the maximum likelihood estimator, which mathematically must be true. So the real question is, take links back to your structural uncertainty. What happens if the simulation mismatches what's going on in the estimation model, i.e. the real world is different from what we think is going on?
Yeah.
I'm not saying you do that for your thesis, but think about that, because that's a really important point to why you show that this model works nicely.
Yeah, it's a good point, and I think it's good to just do, because most people don't even.
I totally agree. Anybody in my
attempt to do
Bayesian class simulate everything. I think that's an incredibly important first step, but I think that's an important caveat.
But yeah, I think that's a good point, because we simulate this is what the population is going to be, and we assume that we know exactly how biology is working out there in the landscape.
Thanks.
Or if I may.
Yeah.
So chapter 2, let me back up. When you parameterize the detection process in your study, what were you trying to describe there? So I kind of think of it as two different things, right? The ability of the camera to trigger if a deer is standing in front of it, that's one element of it. And then the other element is the chance of the deer being in front of the camera to trigger it, right?
Presence, and then probability of it being detected given that it's present. Drawing a blank on, there's one more there. Availability, presence, and then detected given presence. Yeah,
so the reason I asked the question is, I noticed you expected it to vary by year in chapter 2.
Yeah. So
is that because you thought the availability of the deer was varying by year? Is that what you were trying to get?
To be completely honest, is I did all of this with that static binomial mixture model that I used in chapter 1 and put the results up, and it was just super messy. And I thought that variability in the detection probability process was driving some of that noise around the data. So to build on our existing framework and account for that explicitly would help smooth things out of it, and it did.
So you think your really low detection probabilities it wasn't that the cameras were missing deer, right? It was the chances of a deer walking in front of the camera, because you had detection probability below 0.1, right?
Yeah,
Yeah.
they, they're up, they got higher for the chapter 2 stuff. They're like range from like 0.3, 0.5 range, so more in line with what we're seeing with other studies. But yeah, super low, right? You'd think, oh, here's a relatively abundant, detectable species that's literally everywhere. We're gonna put a camera up and we're gonna detect them like crazy. And then it turns out we really don't. Note that these are, not non-baited sites, and they're not on trails. And I think a lot of literature is dominated with trail-based or bait-based. So when you look at it, like, oh, they're everywhere. They're coming in, but we didn't want to introduce any bias there. But we have those two things.
Based on your sampling strategy, then would your camera placement truly random in that way? Like, did you select an area and then randomly position a camera within that area, or were you basing this on some like a priori knowledge of habitat or something? Like this is good to habitats.
Yeah, so that's a good question. The sites were completely random. There's no ground proofing going on out there until you go set up a camera. We had our generalized random tessellation stratified approach where we'd generate a list of sites, and they would be random. And if you work through the sites in that list in sequential order, it would maintain the integrity of a random draw. And that's advantageous in wildlife studies because you're not going to knock on every door and be able to get permission to go on those private lands. So they maintain the randomization that way. And then we went out there and we tried to put them as close to the site as possible, all facing north, like as best we can. If we're out there and it's like, here's this site, oh man, here's someone's bait pile, or here's a nice trail that's like 75 meters that way, we're not gonna go and move the camera to that site. We're gonna stick as close as we possibly can to the random point.
Chat. Last call. Any final questions for Stephen?
Hang out with the pictures.
Yeah, you got a lot of photos here. All right, well, let's give Stephen a round of applause. We'll take a break, and then we'll have the committee come back here in 5-10 minutes or so for the close order session. So thank you all.
If you guys are having this much fun, when's job if your work?
I gotta sneak out every once in a
Are
while.
you proficient in legs? You're a camel at one of those?
I said, it's not your way for just a little advice, not to suck you into something that's going to take a bunch of